Finished looking up at how muscle cells contract, and we're going to look at the role of calcium and how you can get a very rapid muscle contraction in response to a nerve impulse via the actions of calcium. So let's get going with that. So first thing is, most skeletal muscles spend more time relaxed than they do contracted, and so contraction and relaxation must be regulated to produce the coordinated movements which are consistent with normally functioning skeletal muscle. So this is largely under the control, this coordination is largely under the control of the regulatory proteins tropomyosin and troponin, and they can regulate the availability of myosin binding sites on the actin filaments, and they can do that in a calcium dependent way. So we'll look at those two things. We'll look at the roles of tropomyosin and troponin, and then how they respond to calcium. So if we look at this diagram over on the left, and then we'll take a look at this one over here. Here's troponin, TN, and here's tropomyosin, and this is a long filamentous protein which wraps around the actin filament. And troponin is going to regulate the behavior or the position of tropomyosin. So what we see is, if we look at a molecular model of an actin filament, and here is the tropomyosin wrapping around the actin filament. In this conformation, the tropomyosin is blocking the myosin binding sites, so the globular heads can't form cross bridges. Now, if we add calcium to this situation, is what we see is the tropomyosin, in a troponin-dependent manner, slides out of the way slightly to reveal these myosin binding sites. And now myosin can get hold of the actin filament, and we can go through that whole cycle of cocking the head, releasing the phosphate, and doing a power stroke. So what's happening is, here is a thick filament. We're looking down a thick filament, and here's a protruding myosin globular head region. And in the absence of calcium, is what we have is tropomyosin, this thin filamentous protein here. So think about looking down, like we've cut through the middle of this actin filament. The tropomyosin is running along the edge of the actin filament, and here's the myosin binding site on the actin molecule. So that's kind of blocked or occluded by the tropomyosin, and so the tropomyosin is held there by the actions of TNL, TNC, and TNT in troponin. Now, if we dump calcium onto this system, is what happens is that the TNC binds the calcium and has the conformational change, which pulls the tropomyosin away from the myosin binding site on the actin, and now the cross bridge between the globular head of myosin and the actin can form. And so that's the molecular mechanism for how calcium is facilitating the myosin binding to the actin filaments. So when we've got low concentrations of calcium, we're going to be in this state here, or looking like this. And when we've got high concentrations of calcium, we'll look like this. We can bind the myosin heads here, and that's because of this reorganization that we see here in this different view. So is what we're going to ask now is, how do we get these high and low concentrations of calcium in the sarcoplasm, which is the cytoplasm in a muscle fiber, so in a myofibril. So let's talk about how we do that. So what we're going to have is, we're going to have a nerve impulse telling the muscle to contract, but then we're going to allow the muscle to relax. So nerve impulses from motor neurons, those are the neurons which stimulate muscle contractions and things, when they activate particular muscle cells, a nerve impulse is arriving at the muscle, and that causes levels of calcium to go up, and when the nerve impulse stops, the levels of calcium drop. So we're seeing a change in calcium concentration in the cytoplasm of the muscle fiber in response to an incoming action potential. So what we have here is, we have an axon, and we've got all these neuromuscular junctions, which is where the neuron basically innervates the sarcolemma, which is the plasma membrane of the muscle cell. And so is what we have here is a whole bunch of acetylcholine receptors, and so the nerve impulse comes down, the action potential causes a release of acetylcholine in the axon terminals, and that causes a whole bunch of sodium channels to open in the sarcolemma, and what then happens is that there's an influx of sodium into the sarcoplasm, and that causes the membrane to depolarize, and so you have this initial point of depolarization, but is what you have in these specialized cells are these structures called T-tubules, and so you can see all these little holes, and those are long, thin invaginations of the plasma membrane. So the plasma membrane is actually running into and through the muscle cells here, and so these are called T-tubules. And so as the surface here of the plasma membrane or sarcolemma depolarizes, the depolarization can spread down through the T-tubules effectively into the muscle, and so we're allowing the depolarization to spread through the sarcolemma via these transverse T-tubule systems that we have in place. So that inside the cell, the T-tubule system contacts a structure called the sarcoplasmic reticulum, and that's similar to the endoplasmic reticulum, except it stores very, very high levels of calcium. And so when this nerve impulse basically arrives and makes contact with the terminal cisternae of the sarcoplasmic reticulum, that nerve impulse, which has come down the T-tubule, causes a release of calcium by opening voltage-gated calcium channels and using ranitidine receptors and things like that, causes the calcium to exit out of the sarcoplasmic reticulum and go into the sarcoplasm. And so when it goes into the sarcoplasm, it binds onto troponin C, TNC, the troponin C component of troponin, and that causes a rearrangement of TNC such that the tropomyosin is pulled out of the way, and that gives us the access of the myosin heads to the actin site, actin filaments where the myosin heads need to bind, and now we can have that binding, cocking of the myosin head and the power stroke on the release of the organic phosphate. And so when the depolarization is stopped, then we basically have a whole bunch of ATPases which pump the calcium back into the sarcoplasmic reticulum, so we can repeat this process. Just to give you a different look at this structurally, that last diagram is not the most fantastic. Here is the sarcoplasm, sorry, here is the sarcolemma, that'd be the plasma membrane that's wrapping around these myofibrils here. So these are the myofibrils, the contractile units. Here is a sarcomere from this Z line here to this Z line here. And here we have these invaginations of membranes which run into the cell. And so when this membrane depolarizes, say down here, that membrane depolarization spreads through this T-tubule and causes all of this sarcoplasm to discharge, or the sarcoplasmic reticulum to discharge its calcium into the sarcoplasm, and that then causes the muscle contraction that we see because of the rearrangement of tropomyosin and the liberation of those actin bindings or myosin binding sites on actin. So that's as much detail as I think we need to get in for this. Physiology, we'll get into this much deeper for you, but I think that it's important, if you're a cell biologist, that you've got some idea about how some of these things come together and produce a functioning, specialized tissue such as muscle tissue. There's a lot of things that we brought together here, thinking about plasma membranes and thinking about structure and function and thinking about cytoskeleton and thinking about molecular motors. So there's a lot of stuff that comes together when we think about these very specific examples. And if you're going to take physiology, then you'll just be a little bit ahead of the game, having learned this in cell biology. So I'll wrap it up there, and I'll be back soon where we're going to look at microfilament-based motility in non-muscle cells, and we'll do that pretty quickly. I'll see you soon.